My project investigates the role of a long non-coding RNA called NEAT1 in the pathogenesis of a disease called Rett syndrome. Rett syndrome is a severe neurodevelopmental disorder that affects primarily girls, and it's caused by loss of function mutations in the MECP2 or MECP2 gene, which is an X-linked gene. Now, recently, studies have come out showing that both in vitro and in vivo, there are several transcripts that are dysregulated in MECP2 null models. Specifically, one of the most highly dysregulated transcripts is the long non-coding RNA, NEAT1. So we're interested in investigating how NEAT1 can contribute to the pathogenesis of Rett syndrome in various models. I'm specifically working on a cell type called Ren cells, which are stem cells that can be differentiated into cells of the brain. So far in the project, we've analyzed whether MECP2, the protein, binds or interacts with NEAT1, the long non-coding RNA. We're interested in two particular versions of MECP2, the wild-type version, which is unmutated and not implicated in RTT, and the mutated version, in which we've induced a point mutation called R133C in the protein that is commonly found in Rett syndrome patients. So using these two models, we're looking at whether or not NEAT1 interacts differently with each of them, and that can help us understand how NEAT1 could potentially be implicated in the development of the disease. Additionally, I'm interested in the endogenous functions of NEAT1. So NEAT1 is known to be a component of something called paraspeckles, which are subnuclear bodies that harbor various proteins and RNAs important to the cell. So if we knock out NEAT1 or overexpress NEAT1, you can imagine that these various proteins within the nucleus could also be dysregulated. My future studies will involve knocking out NEAT1 so that it's not expressed anymore, as well as overexpressing the transcript to see whether or not those can compensate for any dysregulated processes caused by loss of the MECP2 protein. By overexpressing and knocking out NEAT1, we can later understand whether or not NEAT1 could be a potential target for therapeutic developments. This could be really important for RTT patients because as it stands, there aren't a lot of therapeutics out there that are effective for the disease. Together, we are unstoppable.